All right. Hello, welcome everyone to our first workshop in the R Medicine Conference 2025 on Introduction to R for Clinical Data. I will hand it off to our presenters and we'll get started.
Thanks, Dan. It's really great to see you again. Hello and welcome to the R Medicine pre-conference workshop for Intro to R for Clinical Data. And what I think we'll do is just wait another 2 minutes to allow a few more people to file in, and we'll start at 3 after the hour. 3 minutes after the hour. Panel on the right. So we'll be using the chat window as the primary communication tool to get you help if you get stuck with a technical issue. Please note that the presenter cannot present and monitor and chat at the same time. So if you send me any private messages while I'm presenting, I will probably not see that message, and so I won't respond. Instead, I encourage you to post to everyone, and I think this is the default anyways when it says message everyone down here. Briefly describe the problem you have, and one of the TAs will help you in the chat. I also want to set some expectations for help here. We have 77 participants here today. So please, ask for help if you need technical assistance with Zoom or with the Posit Cloud training environment, or if you run into an error or if you have a hard stop. If you do have a more general question about the material, we encourage you to write it down and ask during the breaks or after the workshop. So we'll be hanging out after the workshop to chat and to talk about some more general things, if you like. So this workshop is interactive. So here's our first interactive exercise. So what I'd like everybody to do is to write where you're dialing in from, your location, city, and country in the chat window. And we'll give ourselves 1 minute to do that. Dan, I'm not seeing any responses in the chat. Are we sure that everybody's allowed to use the chat window?
I'm taking a look.
So Rich was able to say something, but he's a TA.
Uh-oh, hold on, give me 1 sec. I think people can type now.
All right, let's try this again.
Uh-oh. Or I didn't really change anything. Oh, I see.
Looks like in the Q&A chat is disabled.
Yeah. So I enabled that, but there's no wait. Hold on. This didn't happen in the other rooms. Now this is fun. Okay. Try again. There we go.
All right. Okay. So this is fantastic. So thank you so much, Dan, for fixing the chat. So we have Samantha coming in from Cleveland, Ohio, Jonathan from Milwaukee, Netherlands, Germany, Canada, Miami, Florida, Nashville, Tennessee. Fantastic. So we have a guest from Hyderabad, India. That's wonderful. We have a real true international audience here today. That's great. So I'm very happy to see that, and thanks again, Dan, for fixing the chat for us. This is going to come in very useful for the rest of the course. So So this is supposed to be a general introduction to data science and designed for healthcare professionals, clinical researchers who don't have a professional background in programming. So this slide shows sort of a high-level overview of high-level conceptual framework for the most important steps in the data analysis process, and we call it the data analysis pipeline. So typically what happens when you analyze some data, no matter whether, how complicated it is, the first step is to import it into your analytic framework of choice, which in this course is going to be R. Then you start by cleaning or tidying it up. Then you start an iterative process of exploratory data analysis, which we also call the understand cycle. And this usually requires some data transformation. The main ways that you gain knowledge from your data are visualization, making plots, and modeling, creating mathematical or machine learning models. And finally, you need to communicate your results. So we will introduce the basics of all those steps today, with the exception of modeling, because that's a little bit out of scope for this very basic intro course. We will introduce new concept in lecture slides that look sort of like this. And then we'll practice these new skills with timed interactive coding exercises. To do this, we'll have set up a training environment on the web that we specifically set up for you for this course. What I want to emphasize here is that it's important to note that these exercises are timed. And it's not essential that you finish these exercises, but it's essential that you try. And when the time is up, I want to ask that everybody actually stop working and return to the main session, because we'll then live code the exercise to catch everyone up. And I guarantee that you're going to get more out of, watching us reasoning through the exercise than if you try on your own to continue at that point. One more serious thing we need to point out, since this is an interactive course, we want to provide a welcoming and supportive environment for all people, regardless of background or identity. So if you have any questions about this, please take a look at the website, a main website about the code of conduct. And to respect the privacy of participants, please also don't do any screenshots or recordings or photographs while you're in the breakout. Okay, so what I'd like to do now is to do a quick 5-minute meet and greet. So there'll be so I'll try to, or I'll ask Dan to help me send. Actually, let me try to do this myself. I'm going to send everybody into a breakout room where you'll have 5 minutes to meet with 10 to 15 of your fellow participants. So what I'd like you to do is turn on your microphone and camera if you're comfortable, and go around the room and briefly introduce yourself and tell everyone that what you're hoping to get out of the workshop in the conference. And to keep things going, whenever someone is done speaking, they will pass the baton to someone else in the room that they pick. We're tight on time, so it's possible that not everybody will go. And at the 5-minute mark, we'll bring everyone back into the main session. So let me try to open up the breakout rooms. Okay.
I think we need like 6 or 7.
Yeah.
Something on that. 5, 6, 7, one of those numbers.
Yeah, let me see. Huh. Okay. So this is, I just did Are you doing this right now, Dan, or am I doing this?
You can do it unless you want me to do it.
I'd love for you to do it.
Okay.
Since you have more experience.
Give me 1 second. All right. Everyone should have been assigned to a room, and you should be able to see it in the breakout room section below. There we go. We see people disappearing.
Thank you, Dan.
Also, just for your reference, you can send like a global broadcast to give them like
Yep,
a 1 minute heads up. But then when you close it or when I close it, it's still 1 minute. So it's almost like you got to give them 2 minutes.
I see. Oh, that's interesting. I didn't know that. That's useful.
Yeah. Yeah, it catches a lot of people, myself included, when I don't remember when you're like, oh, it's done, but then it gives everyone like a 30 seconds or another minute to like before it actually closes.
That's actually a nice user experience, I think. I didn't look at the clock. Are we like about 3 minutes into this?
I guess like roughly. I also didn't look at the clock, unfortunately.
Okay, that's fine. So if I send a broadcast message now and it says 2 minutes and then I hit close rooms in 1 minute, that's gonna be correct. Is that right? Yeah.
From what I remember, yes, unless they randomly changed it to immediate shutdown.
I don't know. I don't think we need a broadcast message really. I already told him that we're gonna yank him back, so I think that's fine. Okay, I'm gonna send the close all rooms now. And then it'll be all breakout rooms will close in 58 seconds.
Yeah, what ends up happening is some people will see the message and think that you try to get them in now, and then so it always gets a little weird when on both sides when someone clicks close rooms.
Okay. Lecture. Okay, do we have everybody back in the main session? Dan, does it look like everyone's back in the main session? It's tough for me to see from here.
I'm looking. I'm trying to just close all of them now.
Okay.
I think so. I think they're all back. Wait, no, hold on. We have 56. There's people not back. Oh no, they should be back. Because it's closed now. Yep.
Okay. All right. So we'll continue. So in this session, we're going to cover an introduction to R, RStudio, and R Markdown or Quarto. So I'm sure you're all excited to get started programming. We'll do that in just a few minutes. But before we do that, I want to introduce you to three terms. And the first is R. And R is a programming language for data analysis. And there are lots of things that we really like about R. It's freely available. It's great for wrangling data, and it's very capable for producing impressive visualizations. There's also an awesome community of folks of using it for data science, and we're excited to welcome you all to the R community via this course. Then there is RStudio. And RStudio is the name of a free piece of software made by the company Posit, which used to be called RStudio. And you can think of RStudio as a sophisticated text editor for writing R code. And the technical term for this kind of software is IDE for integrated development environment or interactive development environment. I think it's supposed to say integrated. You can run RStudio on Mac or Windows on a server. And for today's workshop, we'll log you into a version of RStudio that's on a server called Posit Cloud. But after today, you'll want to download the RStudio IDE for your computer. You may have heard of Positron, which is an alternative for the RStudio IDE. And at this time, Positron is in public beta, so you can try it out if you like. For the time being, we recommend using the RStudio IDE, though, if you're a beginner. And finally, there's Quarto. And Quarto is a computational document format. And I just realized I have the wrong version of the slides up, so I apologize. Give me just 1 second so I can pull up the correct version of the slides. No, I guess, oh, there we go. Okay. So Quarto is a computational document format that has a executable code inside of it. And we'll look at Quarto very closely in the next shortly. So Quarto also includes capabilities for annotating your work, which is incredibly handy and supports multiple programming languages. So you can mix and match code that's written in R with code that's written in, for example, with Python. You might have heard of R Markdown, and Quarto is the official successor for R Markdown and is very similar. So in R, RStudio and Quarto synergize for a robust ecosystem for performing data analysis. So without further ado, let's dive in and log into RStudio and Posit Cloud. So what I want everybody to do is to log in to type this link into their browser to log into the Posit Cloud environment, and maybe one of our TAs can copy this and paste this link into the chat. There we go. Thank you so much, Rich. Okay, now that the link's in the chat, what you're gonna see is if you don't have a Posit Cloud account yet, click on the Sign Up tab and then enter your email, password, and first and last name. Alternatively, you can also sign up with Google. At this point, you might have to click Create Account. But then you should be greeted with the RStudio workspace, which will deploy like this, and then it should look like this. Once you're on this screen, to make sure your work gets saved, click on Save a Permanent Copy. And if your browser window is large, Posit Cloud will add a top bar and a sidebar. And you don't need these, and you can close them by clicking this upwards pointing arrowhead on the top right of the screen by Xing out Posit Cloud. So here's all the instructions on one page. Log into the Posit Cloud workspace using the link in the chat. Click Save a Permanent Copy, X out of the left sidebar and close the top bar. And then write done in the chat when you're done. And I already see the first done. Raha, you win. Okay, great. Fantastic. It looks like this is working. That's great. So I'm going to leave this up just for another minute or so. Okay. So here's what the RStudio window looks like. On the top left is the editor, and this is where you enter code. And when you've just started R, you may not have that top left window yet, but this will show up as soon as we're starting to edit any documents. On the top right is the environment. Okay, we'll go back to I see there's a few folks in the chat who said they're not ready yet. So when you click Save a Permanent Copy, I just want you to X out the left sidebar and close the top bar to give you more real estate, but this isn't essential. Okay, in the interest of time, I'm going to continue now. All right. So the editor might not be open yet when you first open the IDE, but on the bottom, on the left side, you have the console, and this is where you can quickly run an individual R command, like installing a package. We'll use the console occasionally in this course, but we'll focus on using the editor. The environment is where it allows you to look at the data that's loaded into R. And interacting with data in the environment tab is in some ways similar to working in spreadsheets like Excel. The pane that we've labeled MISC for miscellaneous has a few tabs. The most important one for today is the Files tab, which is the first one, which is activated by default. And this shows you the files we'll be using for this course. So one of the most powerful aspects of working in the R environment is the ability to conduct reproducible data analyses. And those are analyses that can be shared and revised and repurposed and reproduced by others. And those are also analyses where you can start doing where you start by doing something small and then automate it for a much larger production system. To highlight the importance of reproducibility, let's consider the following case. In the mid-2000s, two researchers at Duke University tried to use microarray gene expression data from tumor cells And they used that data to try to predict sensitivity to chemotherapy in patients with cancer. And this approach generated a lot of excitement at the time, and the resulting work was published in some high-profile journals, including Nature Medicine, JAMA, and the New England Journal of Medicine. Unfortunately, there were a number of serious errors in the data analysis, and the media has actually picked up on this, and there was a 60-minute segment. However, that focused on the fact that the researchers tried to cover up investigations into these errors and press on towards clinical trials, even though at the time there were open questions about the validity of these methods. But what's even more unfortunate is that patients were enrolled in clinical trials and allocated based on flawed models. And even though the details were never, published or brought to the public, in all likelihood, patients were actually allocated into incorrect treatment arms. And in the end, 18 papers were retracted, and Duke settled more than 10 lawsuits for an undisclosed amount of money. Keith Baggerly and Kevin Coombs, who were biostatisticians at MD Anderson, were the ones who uncovered these mistakes that led to these distractions, and they did this in painstaking work. Let's look at one of the errors that they found. So what I'm showing here are a few of the hundreds of the microarray probe sets, and each probe set roughly corresponds to a gene. And those are the probe sets that the Duke investigators reported to predict the sensitivity to 5-fluorouracil, which is a chemotherapeutic drug. And here on the right are the probe sets that the MD Anderson team got when they tried to repeat the analysis as described by the Duke researchers. And you can see that the two lists are different. So if I just highlight the last number of each probe, you might notice a pattern. The number of the probe that the Duke reported is exactly one less than the number of the probe that MD Anderson found when they redid the analysis. And this is what's called an off-by-one indexing error. And this is what happens when you use a tool like Excel and you grab one column with a header and one column without the header, and you copy and paste things into a new spreadsheet. And the result is that all the values in the affected column are shifted by one. And this is a fairly simple error to make, but as you might imagine, it completely invalidates all downstream results. This off-by-one indexing error was just one of many simple errors that the MD Anderson team discovered. And another type of error that was pervasive in the study was something called label reversal. And in label reversal is that, for example, cell lines that were labeled sensitive to a drug when they were actually resistant and vice versa. And this type of error can lead to a scenario where a patient gets the chemotherapy that would be predicted to be least beneficial instead of most beneficial. So this is very bad. Other problems they identified were confounding, inclusion from data sources that were not reported in the paper and showing the wrong figures. And, these are all simple errors, and you don't have to be egregiously incompetent or negligent to make them. But because these errors are easy to make, and because without good documentation or reproducible workflow, it's hard to catch them, they're also very common. And a key issue in this case study is that the investigators used point-and-click tools like Excel. And this prevented peers and independent investigators from catching errors in the analysis until it was much later. And the Duke case study is just one example where the critical barrier to reproducibility was the tendency of investigators to use graphical user interface point-and-click type of tools. And these interactive tools usually don't record user interactions. And because of this, they're fundamentally not reproducible. And so we like R because it fundamentally helps us model a reproducible workflow as when we're doing data analysis, but it doesn't just help others. So consider the following three statements and ask yourself if they sound familiar. Number one, can we redo the analysis with this month's data? So this is an important question about automation and moving from ad hoc once-off analyses to something that you want to operationalize. Or Why do the data in table 1 not seem to agree with figure 2? Why did I decide to omit these examples from my analysis? And your closest collaborator is you from 6 months ago, and you can do yourself a huge favor by using tools that promote reproducibility. And if you're analyzing clinical data that might affect patient management or public health based on your findings, that makes it so much more important that you take steps to assure that your analysis is documented, free of errors, and reproducible by others. So, Quarto provides us with features and tools that really tackle the reproducibility problem in a fundamental way. In Quarto, we can craft computer code mixed with narrative annotation that documents the purpose of the code, and you can capture details about decisions that you made in your analysis. And it does this by providing a sort of a lab notebook interface for data analysis, visualization, and annotation of your work. And like our markdown before, Quarto is quickly becoming the gold standard for reproducible data analysis. And this course will teach you Quarto, and we encourage you to continue using it in your future work. And in fact, this is the strongest recommendation that we'll make today, is that you explore using a reproducible document format like Quarto. Another alternative is Jupyter Labs that you may have heard of, but we'll focus on Quarto in this course. So Quarto documents are composed of 3 basic building blocks. And the first is what's called the Quarto header. And it's always on top, and that's required. And this often has details such as the title of the document, which the desired output format, such as HTML, and which editor we're using. So Visual is the default, and it makes editing a little bit easier for novices because the user interface is more like Microsoft Word and less like a traditional code editor. The second type of block is text. And you can have as many text blocks as you want. They just have to come after the header. What Quarto allows you to do is to include some basic formatting, like having different sized headers for different sections. You can have bullet point lists. You can style your text as cursive or bold. You can include hyperlinks, but it's still pretty basic and not as extensive as Microsoft Word. One thing, for example, you can't do is to change the color of your text, at least not easily. The final type of block is called code chunk. And code chunks include R code that can be executed to output results. And to be visually distinct from text blocks, code chunks have a gray background. They're in this gray little box, and they have a different font from the text. So it's easy to see the code chunks in your document. So header, text, and code chunk are your 3 types of building blocks of a Quarto document. So let's zoom in on one of the code chunks. This is super important and may not be familiar to you, even if you've done some programming in other languages, such as Python before. So here we have a code chunk that's written in R. And we know this because at the top of it, it says R, the letter R in curly braces. This tells us the programming language that the code chunk is written. Then there are 2 lines of code. Don't worry about understanding the code for now. But basically what happens here is that we use a function called rnorm to generate 100 random values from the standard normal distribution. And we're taking these numbers and we're assigning it to a variable called x. So x will hold these 100 values in what's called a vector, which is sort of like a list of numbers. Then we call the summary function on x. And the summary function is designed to take a vector of numbers, like the one we just made, and output some statistical summaries, like the minimum, the maximum, etc. And the details aren't that important here. But what I want you to focus on is what happens if we click the green triangle on the top right of the code chunk. So each code chunk has one of these green triangles in your editor. Okay, so what this does is it runs the code chunk in the editor and shows you the result right below the chunk. This can be, so one more detail to note here. The green triangle runs the current chunk only. The symbol on the left of it runs all the code that comes above in code chunks above this code chunk, but not the current one. And this can be useful if you have code above that sets up some data for you that you're then processing in this code chunk. So when you write a document in Quarto, you will use those two buttons all the time. So I want to stop here for a minute and make sure everybody understands this workflow, or if there are any questions, write them in the chat at this time. If you have a huge chunk of code. Yeah, so you can do that. You can have one humongous code chunk and write all of your code in there. That makes it, we don't recommend that. We recommend having code chunks being pretty small, and each code chunk only do a few, only really do one a very small piece of the analysis. And this way you can better understand what your analysis does. You can write more descriptive annotations between the chunks, and you can run them independently and iterate faster. So yeah. Does Quarto have support for mathematical formulas such as LaTeX? Yes, you can write LaTeX formulas in Quarto documents. We're not going over this, but that's easy to look up, and you can definitely do that. So at some point, you might want to create a human readable, shareable version of your document, and this is called rendering. And you can render the document by clicking on the render button. And you can see that the output is a neatly rendered document with the code and the results shown. So let's zoom out a little bit now and look at this Quarto document in greater detail. So we have a review. So we have the header section up here. The second the second block is a text block. That's here, starts with large header and has some style text in here. Then we have a code chunk. This code chunk sets up the X vector and outputs a text summary. And then we actually have another text chunk, or a text block, which just says including plots. And then we have another code chunk here. And this one invokes the hist function to create a histogram. One super important thing to note here, so this hist function is invoked on the X variable, but we defined the X variable up here. So how does this work? So I can use the X variable I defined above in the first code chunk in that second code chunk. And this is because when I'm working in the editor on a Quarto document, R is running in the background and keeps track of the variables. And when I render a document, it starts a new R session, and the R session runs all the code chunks in order. And so it remembers all of the data that we define during in between the code chunks. So this really lets you break up an analysis into small independent pieces that are easy to reason about. Okay, it's time to practice. So what I would like you to do is, so basically what we're going to do now is we're going to create a Quarto document. We're going to review the format. We're going to execute some code chunks and render the document. And so I'll just read out the instructions, and then I'll leave this up for a couple of minutes. This shouldn't take you more than a couple of minutes to complete. But basically what you want to do inside of the Posit Cloud environment, click on File, New File, Quarto document. This will ask you to enter a title. So you can enter R Medicine 2025 for the title, and then click on the Create button. Now an editor window will open up with your Quarto document. And R is so the RStudio IDE will actually, when you create a new Quarto document, it's not a blank file. It'll pre-populate with some example boilerplate, and we're going to look at that boilerplate. So importantly, the Posit Cloud environment doesn't actually let me pre-install packages for you. So you'll get a little warning that'll say that you need to install R Markdown. Just click on the install link that shows up when that happens, and then once you've installed R Markdown, you should be able to do the rest of the exercise. Look at the format of the document, identify the header, the text blocks, and the code chunks, and then execute each one of the individual code chunks by clicking on the green triangle. Then when you're done with that, try to render the document to HTML. When you're done so, when you do that, it'll ask you to save your Quarto document first, and you can give it a file name such as test and click save. You might get a pop-up about opening an external browser. This is you just click try again then, and then you should be able to see the rendered document. And if that was a lot of information, just try your best at following these instructions. We'll come back together in 3 minutes and work through it together. Oh, and if you're done, if you're able to do everything, just write done in the chat to let me know. Wow, you guys are fast. Okay, let's look at this together real quick. So if you're still trying to work through this, please stop and watch me instead. So sometimes the deposit cloud kind of goes to sleep, so to wake it back up. Okay. So we're opening up a new Quarto document. And I'm going to call it R Medicine 2025 and click create. And it didn't ask me to install anything, which is fine. That's great that it didn't ask you to do that, because it can sometimes take a little while for packages to get installed. So we'll have the header up here, and then we have our first text block here, a code chunk, a text block, a code chunk, and another text block. Execute each individual code chunk. So we're clicking the green triangle here, and this code is asking R to calculate the result of 1 + 1, and we're getting 2 as the response. You can ignore this bit before the result. And if we run this code chunk, 2 × 2 is 4. And then this down here says that the echo false option disables the printing of code. Of course, we see the code printed inside of the editor, but when we try to render the document, I'm just going to call this test. So it just saved a test.qmd file. And here I get my pop-up block alerts. I'm going to just hit try again. I get my HTML document. And there was a question about what is the best document to render into. I think that it really depends. HTML usually looks the nicest, and you can save the HTML file and email it to your collaborators. But other, but PDF is fine too. And we'll talk about other formats that you can render into. So the default is HTML. So one of the questions was, if you look at the rendered document, did the first code chunk show up? Yes, code chunks get included in the document by default, but maybe you don't want to confuse your collaborator with all the complicated code for an analysis. And then you can also hide it. So the second code chunk, we actually excluded the code itself, and we're only showing the result. And we did this by including what's called a chunk option. And here the chunk option is echo false. And you can look up chunk options for Quarto. There's many of them, and I don't want to go into detail about them at this point, but there's plenty of ways for you to customize what your Quarto document is going to look like. Okay. Now that we looked at how to create Quarto documents, we can begin the process of performing data analysis in earnest by importing a clinical data set. So we looked at this data analysis pipeline in the welcome presentation, and the first step in the data analysis is to load or import data into the environment so we can begin to tidy and transform that data, and then later visualize it or model it and communicate it. So in today's course, we'll be using a de-identified data set de-identified synthetic data set consisting of COVID-19 lab test results from a microbiology lab. And this data is stored as what's called a CSV file. So what's a CSV file? CSV stands for comma separated values. And a CSV file is a plain text file. Which means you can open it in any text editor and look at it. Here we have a CSV file with the names, medical record numbers, and dates of birth of our 3 fictional patients. This data structure is also is called rectangular because it falls into rows and columns, where each row has the same number of columns. So 3 columns, name, MRN, date of birth, and each column has the same number of rows. Okay. Also note that this particular CSV file has a header row that instead of data has a descriptor of what kinds of data are found in each column. So this says MRN, which isn't itself an MRN, it's a descriptor of what's going to be in the rest of this column. So CSV files often have a header row like this, but they don't always have a header row. To import our CSV data, we need some additional data analysis tools. In this course, we will be leveraging the tidyverse. And the tidyverse is a modern set of tools for data analysis in R. It's quickly becoming, or it's really the de facto standard for doing data science in R. There's some basic tenets of tidy data analysis, and they include that data should be organized in a consistent, standardized way, where each row is an observation and each column is a variable, just like in our CSV file just now. This is a very common way to organize data in a spreadsheet. So this should be kind of intuitive. The next tenet, another tenet of tidy data analysis, is that programming code should be human readable and concise and consistent. And as much as possible, it should mimic narrative English language as possible. And this is sort of an R thing. We don't usually see this in other programming languages as much, or Python and in JavaScript definitely don't have this human readability as much in mind as R, but especially tidyverse R is trying to be human readable. And this is why we like, it was a big part of why we like it. And then finally, each data analysis should be possible to be broken down into a series of atomic data analysis steps, such as select this column or arrange the data by the values in that column. And accordingly, you should be able to put together an arbitrarily complex data analysis into a pipeline of atomic steps. So the tidyverse is a package, and a package is a collection of functions, of data, and help documentation, all in really like in a sort of a zip file. And you can download this and install it into your R environment to extend its functionality. So you need to install packages explicitly with the command install.packages. So let's say you want to install a package named tidyverse. You can go to the R console and type install.packages, parentheses open, and tidyverse in quotes. And these can be single quotes, double quotes. In R, single quotes and double quotes are the same thing. So each package you want only needs to be installed once on each computer. However, in order to use the functions or the data in the package, you also need to load the package. So R doesn't automatically load all the packages you have installed because that would be a waste of memory, because probably you don't need to use all the packages in your specific script. And so this is why you need to explicitly load your packages. And this is done with the command library. So to enable all the functions in the tidyverse package, you type library parenthesis open, quote, tidyverse, quote, parenthesis closed. And packages remain loaded until you quit the R session. Every time you start a new R session, you have to load each package that you want to use again. If you're writing a script and you're running a script, and you have to type, you write the library as one of the instructions early up in your script in what we usually call a setup code chunk or setup chunk. So we'll see that later. On Posit Cloud today, you might be prompted to install some missing packages with a warning that looks like this. Earlier on, I've gotten feedback that this wasn't necessary, but I don't know, this happens sometimes. If this happens, you can click on the install link when it appears, and that'll start the process of installing. So these are the two first R commands we covered: install packages in the library. So let's actually go ahead and install the tidyverse. This is going to save us some time later on when we're doing exercises that use the tidyverse package. If you already have it installed in your environment, it doesn't hurt to install it again. But if you're installing for the first time, it might take about 5 to 7 minutes. So what I want you to do is to go to your console, type install.packages, parenthesis open, quote, tidyverse, quote, parenthesis closed, and hit enter. And type done in the chat, not when the installation is finished, but when the installation is started and you're seeing that it's doing stuff. Okay. Okay, I'm seeing a lot of dones in the chat, so we're gonna move on. So let's say you just loaded the tidyverse package using the library statement. You can import CSV file using a function called read_CSV. And here is a template for how to use the read_CSV function to create a data frame object from a CSV file. So you start with the name of the object that you want to have in your environment. And this is going to be called a data frame object. We talked about a vector object earlier. Data frame is a different kind of object. And this is an object that captures all the contents of your CSV file. So you start with the name of the data frame object that you want to create. Then you have this leftward arrow looking thing, which is a smaller than followed by a dash, a minus sign. And then read_CSV, and then the file name in parentheses. This code construct is exceedingly common in R, so I want to spend a few minutes exploring it together. So read_CSV is a function. And remember that functions are defined in packages, and we loaded the tidyverse package to be able to use the read_CSV function. So you may be familiar with functions from math class. A function is something that takes an input, say an X value, and returns an output, say a Y value. And functions in computer programming also take inputs and return outputs, but the inputs and outputs here are arguments and objects that exist within the context of a programming language. And for read_CSV, the input is the file name of a CSV file, and the output is a data frame with the contents of the file. So the input that goes into a function is called an argument. The argument to a function gets always gets put in parentheses after the function name. And a function can have 0, 1, or many arguments. And if there's more than one argument, we use commas to separate them, and we'll see examples of that later. The read_CSV function outputs a data frame. So think of a data frame as just like a table, like what you have in an Excel sheet. But if we want to capture that data frame inside of a name object, we need to specify that explicitly. And it's usually a great idea to capture the output of a function in an object, so you can use that object as input for other functions, for example, to summarize or visualize the data name object. So to do that, to put the output of the read CSV function into a named object, we use what's called the assignment operator. And the assignment operator is a smaller than followed by a dash or minus and kind of looks like an arrow pointing left. So this assignment operator, again, takes the output from what read CSV, a file name, generates and then creates a new object with a name that you specify, which stores the output. And we usually, when we read code out loud, we usually pronounce the assignment operator as gets, as in data frame gets read CSV off file name. And it can be useful to think about how you're reading code out loud when you're discussing code with your collaborators. So let's put these pieces together to load our COVID data set. So in this line of code, this kind of line of code reads, COVID testing gets read CSV of COVID_testing.csv. And you might notice that one of the objects is in quotes and the other one isn't. And to be honest, quotes can be kind of confusing in R and in programming in general. Just some general rules are that names of objects that you're using in your code don't get quotes. If you're referring to a file name in your file system, one of your folders, that always gets put into quotes. And this is just, part of the grammar of coding, like the syntax in R that it just takes time to deal with, to become familiar with as you develop proficiency and comfort with the language. But this code here, again, COVID testing gets read CSV of COVID testing.csv will generate a COVID testing data frame, which we can later then use to display and transform and summarize and visualize and model. So this is pretty powerful. So let's do one more your turn in the session. And in this exercise, I'll ask you to open a Quarto document and follow the instructions that I've written in that document. And those instructions, so you're going to work inside of a Quarto document to load and explore a data set. And this is going to mimic sort of what you would do as a data analyst. So go to the bottom right pane, the files pane, click on the exercises folder, and then find in there a Quarto document called 01-introduction.qmd. QMD stands for Quarto Markdown. And what should happen is that in your editor pane, this file should load, and the file is going to have instructions to complete the exercise. And we're going to give ourselves 5 minutes. In 5 minutes, we'll come back. We'll go over this exercise together. And if you're done earlier than in 5 minutes, feel free to hit done to write done into the chat. And so Shiva is asking for a GitHub link to the QMD file. Rich, maybe you can help her with that.
Sure thing.
Thanks. Okay, everybody, let's come back together and work through this exercise together on my screen. So I'm opening up the introduction.qmd file, which pops up right here. And it tells me about the COVID testing.csv file that I can see here. I want to import it into R as a data frame and look at its contents using the data viewer, which is one of the most awesome features of the RStudio IDE, and also part of the reason for why I don't at the moment recommend Positron, because its data viewer is not as good as the one that's in RStudio. So we'll run the following code chunk, which is gonna load the tidyverse, and we need the tidyverse to unlock the read CSV function. And read CSV is gonna look at the COVID testing.csv file and push it into a data frame named COVID testing. So right now I just cleaned up my environment. Right now my environment is empty, and after I run this code chunk, this COVID testing data frame appears here. So the environment is another really cool feature of the RStudio IDE, which doesn't really have a good equivalent in Positron right now. And in that, it's always very easy for me to see what the current status is of all the data that I have loaded into my R environment. So I'm going to just ignore that stuff here. R tends to be pretty verbose sometimes, and tells me stuff that I didn't necessarily ask for, and that's okay. All right. So let's jump to the questions. So how many rows are in my data frame and how many columns? And there's a couple of ways to know this. The first is that right here in the environment, it tells me that we have 15,524 observations, OBS dot observations of 17 variables. Now, observation is the same thing as a row, and a variable is the same thing as a column in a tidy data set. But the other thing I can do is I can click on this table button here to open the data viewer. And now I see that in the bottom of here, it tells me that we're seeing 15,524 entries and 17 total columns. So we have 17 columns, and this is how many rows we have. Okay, so there's two ways of doing some of knowing some very basic important information about my data frame without writing any code. All right, go ahead and try to edit any of the values in the viewer. It would have been easy for RStudio programmers to allow editing, but why did they decide not to add the feature? And why do you think this was designed that way? What did people think? Would anybody volunteer to venture a guess in the chat? No reproducibility, Shiva, you got it exactly right. If I can, do my analysis in here and go into the data viewer and start messing around in it, and then later on, I published that, and I published an analysis, and somebody tries to do it again, there was no recording of me editing anything in here. So this is as of by design not allowed. And there's a big difference to Excel, where you can absolutely mess around in the spreadsheet. So each row in COVID testing represents a single COVID test. The value of pandemic day indicates the day on which the sample was taken, on which pandemic day was the first sample taken, and the last. So I can look at this by clicking on the on these sorting arrows here, and I see that the first sample was taken on day 4 and the last one on day 107. So I can easily use the data viewer to sort my data frame. So one important thing to understand is that we're not actually changing the data in the data frame. This is just a view of the data, but my COVID testing data frame still is in the same order and wasn't changed. So this is another big difference to how Excel works. Okay, and how many tests overall were positive? How many positive tests were there in the first 30 days of the pandemic? And without that hint, you may have been led to think that, okay, now it starts now it starts, we're gonna have to start to actually writing some code. But the data viewer includes a filtering function. So so what we can see is that the result column tells us whether a test was positive or negative. It looks like invalid is another option, another possibility. But I can, oops, that was not supposed to happen. All right, here we go. I can click on the filter button, and now under each one of the column headers, I get to, I have this extra text field. I can write part of the string that I want to, or part of the result or the value that I want to filter for. And now it's filtering things down. It's filtering the table down to only the matches. And down here, it tells me that now we have 865 entries filtered from 15,000 total entries. So this is incredibly handy. You can start doing your exploratory data analysis with the data viewer without doing any coding, and that's really wonderful. Here we can, I think the question also asks us to filter things down in addition to the first 30 days, and now we are at 137 entries, and I believe that is the correct answer. Okay, all right. So let's recap. So we have we talked about packages, and packages extend the functionality of R. They need to be installed once per computer and loaded, but loaded for each session. And then we talked about functions. Functions do stuff. They accept arguments, and they can output objects, and we can store the output of functions using the assignment operator. And importing data is the first step of the data analysis pipeline. If you have CSV data, you can use the read_CSV function from the tidyverse for importing data. And the tidyverse provides functions to import other kinds of data as well. But CSV is very common. So at the end of each one of these lectures, we'll have a brief what else section to introduce you to other things you may want to explore and learn about after completing the course. But we're not going to go into any great detail. And so this includes some helpful hints, other functions of the tidyverse, and packages that you may want to check out. So that a lot of R packages, especially those maintained by the POSIT company, come with cheat sheets, and the data import cheat sheet helps with the grammar of importing data, including other file types and those with other separators, for example, pipes instead of commas, which is not entirely unusual in clinical data. The data import cheat sheet, you can find it on the POSIT website. There are lots of other packages designed to deal with different file formats. To read Excel files, there's the read Excel package. Google Sheets 4 for importing data that is stored in Google Sheets. JSON Lite helps with JSON data and APIs on the web. You can use Haven to import SPSS, STAT, and SAS files, and REDCap Tidyverse to easily import REDCap data. So outside of just files on a computer, you can also connect to many different databases directly in R to source your data. And if you use any of these, you may want to check out the tools to make database connections in R, which is linked down here. Actually, I think I need to update that link, but you'll be able to figure this out from the POSIT website. In this lesson, we practiced rendering our analysis into HTML, but the question came up what other formats are out there. PDF is one of them. You can render into Microsoft Word, PowerPoint, but you can even render into interactive dashboards. So we'll actually look at dashboards in greater detail later in the lecture, but the key thing is that Quarto is sort of at the center of all of this, and you can in the end deploy you can communicate your analysis in many different ways. Finally, I want to highlight the Reticulate package, which provides an interface between R and Python. And this means you can mix code that are written in R with code chunks written in Python. And R does many things well, but no programming language is perfect for all tasks. So it's great that R gives you the tools to mix and match languages in different code chunks. And but for those of you new to coding, I recommend focusing on a single language to develop proficiency before moving on to another language, and this will shorten the learning curve and with your first and future languages. Okay, this is the end of this session. So what I want to do is, because I want to take a quick bio break. So let's get back together at 35 after the hour. So so that'll be in 10 minutes. Thanks for pointing out that my microphone is muted. So thanks for that, and welcome to the second session on data visualization. Let's take another look at the COVID testing data frame we looked at earlier. So you might remember that it had lots of rows and columns, and each row represents a single COVID-19 lab test in this synthetic data set, where the pan day variable represents the day of testing, starting with 0 at the beginning of the pandemic, which was sometime in late February of 2020. So by just glancing at the first couple rows, you can see a few things right away. There was only one test on the 4th day, then 2 on the 7th, and 3 on the 8th, and then a bunch more on the 9th. So it seems like there's a gradual ramp up in testing. Also, you can look at the result column, and it looks like all tests early on were negative. So now I'm going to ask you to imagine what the following plot might look like. So if you take the COVID testing data frame and you plot on the x-axis the pan day, the day of the pandemic, and then the y-axis the number of tests that were performed on that day. So just take a few seconds and try to visualize this graph in your mind or doodle it on a piece of paper in front of you. And I want you to just take a few seconds to think about this. And once you have an idea, just write done in the chat. Okay, everybody have an idea? So what I've asked you to imagine is a plot in which we have the count or the frequency of a test on the y-axis, and that's then broken down along the pandemic day over the x-axis. Anybody know what the name of the type of plot is that we where we have a thing, a count of the thing on the y-axis and the distribution of those counts along the x-axis? And that's exactly right. That's a histogram. So let's build a histogram of COVID tests by pandemic day. So what I'm going to ask you now is to go into RStudio console and type in the code starting with ggplot exactly as it's written here. And pay attention to the spelling, capitalization, and parentheses. This can be a little bit complicated. But type this command into your console and type down in the chat when you're done and you see your histogram. I just want everybody to get a little bit of muscle memory of what it feels like to write a ggplot command is the point of this exercise. Okay, Martine is done. Thanks for letting me know. Okay, I'm seeing a bunch of dones in the chat. So is this what you get? If so, that's great. Actually, when I run this code, I get what looks like an error, but it's actually just a message. And R lets you know here in this cryptic sounding, response that it was asked to draw a histogram, And when you ask it to draw a histogram, you should tell it how wide each bin should be, because this affects the granularity of the data displayed. And this is actually a useful message. And we will later on how to look at that. But I also wanted to point out here that sometimes R will put out this fire truck red stuff. And if you're a beginning R user, you might feel like you did something wrong because this looks like an error because it's red. But just because something is red in R doesn't mean it's an error. Actually, when you get an error, it will always say error. So this is just a message or a warning. And I just wanted to point that out because this can throw people off. So why did we do this? So when I asked you, why did I imagine you what the plot looked like? So what we're seeing here is the number of COVID tests that were performed on a given day over time. And you might have imagined that it looks like something like this. Initially, you have very few tests, and then it ramps up. As the test becomes broadly available, and at some point, the number of tests hits a plateau. And so you might have imagined something that looks like this. But this graph, this visualization tells you so much more than that general shape. You can see that by 30 days, we're hitting that plateau. And it also tells you a few things that you might not have expected that you might want to look at more, like there's something going on with the testing volume fluctuating up and down. The thing is that none of this would have been apparent from looking at a table with 15,000 rows. So the point of this exercise is to realize that you might have some ideas about your data, but usually they are incomplete and oftentimes they're wrong. So if you don't have a way to quickly visualize the data, you're probably missing important information. Being able to visualize data quickly is critical for understanding. And it might sound trivial, but it's actually profound. And it's why we call visualization as one of the two main engines of knowledge generation from data, the other one being mathematical modeling. But this is why we're emphasizing visualization so much in this course. So we'll be using the ggplot2 package for creating graphics. ggplot2 is part of the tidyverse, so it'll get loaded when you load the tidyverse package. ggplot provides a grammar of graphics. That's what the GG stands for. So it's a grammar of graphics for data visualization. And the idea of having a grammar for something is actually pretty common in R, and we'll revisit it in the session of data transformation. Essentially, the idea is that there should be a consistent way to build any type of graph. So you shouldn't have to learn how to make a histogram, and then there's a completely different way to make a line graph. So just makes it to have a grammar should make it easier for humans to learn how to make graphics and also how to read the code later and make sense out of it. And also it should make it easy to create a whole bunch of different graphics to look at different aspects of the data. So the idea of the specific idea of the grammar of graphics is that you should be able to specify any graph by specifying three things. Number one, the data that goes into it. Number two, the type of graph that you want to make. And number three, a mapping that describes how the data should be represented as visual marks on the plot. And so having a consistent grammar like this means that once you learn how to make a histogram, you can apply this knowledge easily to make a scatter plot with little extra effort. And this makes it easier to generate lots of different graphs quickly and help you understand your data more quickly. So also ggplot2 graphs look awesome, and you can generate publication quality plots pretty straightforwardly with ggplot2. So here's a quick analysis of how we just use ggplot to make a histogram. You can see that we give it a data frame, in our case, COVID testing data. This is what we pulled out of that CSV file earlier. Then we specify the type of plot, or geom, in this case, a histogram. and we specify an aesthetic mapping. In this case, we are saying that we want the x-axis to represent the pan-day variable of COVID testing. And since this is a histogram, I don't have to specify the y-axis, because in a histogram, the y-axis is always the count of cases, which for COVID testing is the count of tests in a particular bin. So a couple more. Okay, so we have a data frame, we have the type plot and the x-axis mapping. So these were the three pieces in the grammar of graphics that we say we need to specify to make any kind of plot. A few more little details. You always start a plot with the ggplot function. And to connect the ggplot function with the geom function, we use a plus sign, and oftentimes we write that at the end of the line. So there's a new line after that, just for better readability. And the mappings go inside a function called AES. And AES is for aesthetic mapping. So that was a lot of information all at once. Let's try to consolidate this into a more general template. And here is the official template for making any kind of graph with ggplot2. You start with the code written in black, that's your constant part, and then you fill in the details in blue. The first detail is a tidy data frame. And this contains the data that you want to plot. So what do I mean by tidy data frame? So data set can take on a lot of different shapes, but there's one shape that is best suited for data analysis, and that shape is called tidy. And a data set is tidy if each variable is in its own column, each observation is in its own row, and each value is in its own cell. The opposite of tidy is often called messy, and oftentimes a lot of the data analysis work is to convert messy data into tidy data. But for now, fortunately for us, the COVID testing data set is tidy already. So, so we have our tidy data frame. The next step is to pick a geom function. And a geom function tells R what kind of plot you want to create. So we'll go into more detail about geom functions R, but for now, I just want you to know that you need to tell ggplot what graph you want, and you do that by picking the right geom function. And here, so the top 6 most useful geom functions for visualizing clinical data, but there are many more. And with these 6, you can make histograms, bar plots. Geom point is for scatter plots, dot plots, box plots, and line graphs. So now we picked a data frame, a geom function. The third and last and the least intuitive step is to write aesthetic mappings. So what's an aesthetic mapping is basically how you tell R how you want the columns of the data to show up on the plot as graphical markings on the plot. So what's an aesthetic and what's an aesthetic mapping? An aesthetic is a thing that we can perceive about a data element, about a specific individual data element on a graphic. And this is, not just things that are generally on the plot, but things that are specific for each row of each observation of a data set. So this could be things like a position on an XY grid, but it's more general than that. It could also be the color of a line or of a dot, if those colors vary in between different cases, in between different observations. And that's what an aesthetic is. An aesthetic mapping is how you tell R how you want to represent the columns or the variables of a data frame on the plot. For example, here we have a data frame with 3 columns, A, B, and C, and we have this XY grid. The aesthetic mapping above defines that the x-axis position of each marking should be derived from the A column, x equals A. All right, so we're going to put whatever says A as the x position. We want to map the B column to the y-axis, so whatever the number in B is going to be the y position, and that the color should come from C. So here you can see how aesthetics generalize just, X and Y to more different attributes, more different things that can be perceived about a specific data element on a plot. So as a result, you get the following graph, and that following graph completely visually encodes all of the information given in those 3 columns of the data frame. And R automatically figures out reasonable axis limits in the color scale, but you can also manually fine-tune this. So aesthetics are a powerful and fundamental concept in the grammar of graphics. So let's do a quick your turn to solidify this. So in addition to X, Y position and color, what other aesthetics can you think of? And type your answers in the chat. Shape, okay. Size, yeah, perfect. Okay, text. Oh, we can talk about that. So it depends on what you mean by text. Stroke, fill, okay. Alpha, that's a great one. Okay. So there's actually a bunch of different things, and they also, it's important that the kinds of aesthetics that are available depend on the plot you're making. For example, for a line graph, you can define the line width and the line type, but you can't do that for a scatter plot. On a scatter plot, you can define the shape of the dots, and so picking the best aesthetics for your graph is as much an art as it is a science. And I actually do recommend Klaus Wilke's Fundamental of Data Visualization for a great introduction into this topic. And I'll have a reference. I'll have a link to that at the end of this presentation. So let's recap. To make any kind of graph, you start with this template and you choose a tidy data frame. And this contains the data you want to plot. You pick a geom function, and this is the type of plot you want to create. And you write your aesthetic mappings. And this is where you tell R how you want to map the data columns to X and Y axis and color and whatever else other features of the graph. Okay, so let's do some exercises. We're running a little bit tight on time, so we'll probably only do the first half of the visualize QMD, but work through the exercises of the section titled Your Turn 5 and then stop where it says stop here and type done in the chat when you're done. We'll give ourselves 5 minutes and then come back together and look at this together. Okay, I see a lot of dones in the chat. Let's look at this exercise together. Okay, we're loading tidyverse and lubridate. Lubridate is a package that allows us to deal with dates, and we're loading the COVID testing data frame. Okay, so here we have the template again, and we want to plug in COVID testing as the data set, geome histogram as the geome, and map X to Panday, very similar to as in the in my slides. And so this is a sort of fill in the gap kind of exercise, so I can start filling things in. So one thing I want to point out is something that just happened as I started to type the COVID testing data frame into the editor. And this is something that's called autocomplete. And it's worth mentioning because if you're not familiar with coding, this is a powerful feature that I encourage you to use all the time. So basically what happened is that the RStudio editor realized that I'm typing something that it recognizes that the RStudio IDEs basically tell me, hey, I think I know what you're talking about, and it gave me this little box in which it actually just gives me some information, a preview of the data in here, and what I can do. I can start typing and I can complete this by hitting the tab button. And this way I don't have to spell out um, the full name of the object, reduce the chance of making a typo. And so autocomplete is great. Okay, geome histogram. So you may have wondered why like the different geome functions all start with geome underscore. One of the reasons is autocomplete. And here's something cool that happens when I start typing geome. I'm actually getting a list of all of the different objects that are loaded into R that start with geom. And not only does that help me finish what I'm typing, but it also helps me discover uh, discover functionality. So here I get an overview of all of the different geome functions that there are. But for now, I know I want the histogram, so I'm just going to keep typing enough letters until it's unique, and I can hit tab. The other thing I want to point out is that next to the autocomplete box, we actually see the beginning of the documentation for this function. So here it tells me all the different uh, arguments that it'll accept, and it'll start telling me sort of the first the main so some of the key concepts about how to use this function or what this function does. So I can hit tab again and be done with auto completing here. All right, so we want to map X to pan underscore day. And unfortunately, autocomplete isn't smart enough to look inside of data frames for aesthetic mappings, but oh well, can't have everything. All right. So we hit the run code chunk icon, and And we get this stat bin using bins equal 30. Actually, it shows up in a separate window, but it does tell us pick a better value with bin width, and this is actually a good idea. So let's do it here. And you may have noticed that I'm making you type the same thing multiple times. And this is really just for your benefit to get some muscle memory in typing some of these ggplots. I'm not doing this to torture you. Okay, so what is the correct bin width? What did you guys find is the right bin width for seeing daily test volumes? Anybody want to write it in the chat? Okay, 1. And why is it 1? Yes, exactly. We want to look for daily test volumes, and pan day is the pandemic day. So bin width of 1 will give us the daily test volume. Exactly. All right. Okay. So now this is a lot more granular. So anybody notice that there's these peaks and troughs in this testing data? Anybody want to venture a guess as to what these what this might represent? Okay, Hajira says weekends. So that's a great suggestion. And oftentimes when we look at clinical data or lab data, we'll see weekly variations because typically the lab runs much fewer tests on the weekends. Whether that's a good thing or not, that is just the way it is. So you're exactly right. And you could confirm this by counting up, by ensuring that you're gonna have exactly 7 that these peaks and troughs happen at a cadence of 7. But that's great intuition. So we stopped here. Now unfortunately, we're running low on time for this workshop, and I want to make sure that you guys at least see some of the transformation and some of the dashboarding stuff we have in our slides. So these slides are going to be available for you, and the teaching environment, the Posit Cloud environment is going to be available for you after this course. I just wanted to, but I'm going to go to the rest of this lecture really quickly. So setting versus mapping aesthetic, we talked about mapping aesthetics. Setting aesthetics basically refers to making the whole entire plot a specific color versus going by a specific taking the color from specific rows of data, and so I'm going to skip forward to the recap, and I'm going to highly recommend that you guys check out the exercise, the rest of the exercise from this QMD after the course. But now to summarize ggplot, it's a package that provides a grammar of graphics. You can create any type of plot using a simple template to which you provide a tidy data frame in which each variable is a column, each observation is its own row, and each value is in its own cell. A geom function which tells R what kind of plot to make, and aesthetic mappings which tell R to how represent data as graphical markings on the plot. We did not go over how aesthetics can be set to constant values, but we went over how aesthetics can be mapped to a variable. And I encourage you to go into the exercise after the session, after the workshop is over, not while the workshop is ongoing, to delve a little bit more deeply into to practice some more mapping of aesthetics. So what else is there? We really only scratched the surface of what ggplot can do a tiny little bit. And I want to point out that you can manually save. I want to point out a few things. The first is oftentimes you want to manually save a plot image to put it into a PowerPoint slide. This is not necessarily the reproducible thing to do, but you can do that. You can go to the plots pane, which is in the MISC pane, and click export and select save as image. If you want to save a plot that's by running some code in an R notebook, you can just right click and click on save image as. So those are different ways of manually saving plots. In ggplot, you can really customize. There's a lot of customization options. For example, you can adjust the position in which overlapping objects are arranged. For example, you can instead of a stacked histogram, you can have side-by-side bars. You can use different themes which affect how non-data elements such as axes, grid lines, and background appear. So here we have a different theme that makes the grid lines a different color. You can customize the color scales. You can facet a plot into subplots. You can use radial coordinate systems, or you can use maps, geographical maps to create choroplaths. And you can add titles, subtitles, annotations. You can change your axis labels or the appearance. And all of this, the syntax to do all of this stuff is you still start with your basic template, and then you can add another plus and add a theme function and plus a scale function, or a whole bunch of other functions are available to keep modifying the plot. And this is really handy because it allows you to sort of iteratively configure and refine your plot until it looks the way you want it to. The ggplot cheat sheet, which you can find on the POSIT website, is really neat to have on hand as you're exploring your data. Here you can see, first of all, you can see that the template to build a graph. You can see I didn't make this up. This is canonical ggplot teaching, and you can see some of the main geom function for one variable and two variable plots. If you'd like to learn more about which graphics are most effective in specific situation, I recommend looking at Fundamentals of Data Visualizations by Klaus Wilke. This is a very readable primer on data visualization and figure design, and you can read it for free at the address shown on the slide. And I believe Rich posted that link in the chat earlier today. There's a lot of packages that extend the ggplot basic functionality for more complex or for more specialized purposes. We recommend the ggsurffit package to make survival curves and risk tables. There's a few packages we recommend ggsurffit because it sticks pretty closely to the grammar of graphics and the way ggplot is meant to be built. And it's also written by our good friend Dan Sjoberg, who is chairing the R Medicine conference this year. If you want to create swimmer plots, which are popular type of graph in the medical literature, please do take a look at the ggswim package written by Rich Hanna, my co-presenter. And you might also want to check out Rich's lightning talk on Friday at 3:34 p.m. Eastern time, I believe. And finally, the GT summary package makes it super easy to generate publication-ready display tables from a tidy data frame. So so these are some of the packages that I encourage you to check out. So at this point, I'm going to hand the reins over to Rich for the next session.
All right. Give me 1 second. Okay, can we see the slide?
Yes.
Great. Just 1 more second. Okay, thank you. So hi everyone. Yes, my name is Rich. I'm a data scientist here at CHOP. It's my pleasure to give Stephan a little break and take you through the transformation portion of today's workshop. As Stephan mentioned earlier, I cannot see the chat right now, so hopefully everyone else can weigh in on any questions. So this is the generalized data science pipeline that was introduced to you earlier today, and the functions we're going to learn about fit specifically within that tidy and transform portions of the workflow. So we'll be using functions that are part of the dplyr package, which is a sort of a portmanteau of data pliers. The dplyr package is part of the tidyverse meta package and a workhorse for data transformation. I use it all the time in my day-to-day work, and honestly, if I had to pick a package that was the most critical to my work, it's probably this one. And dplyr itself isn't just a haphazard collection of functions. It takes a holistic and almost philosophical approach to the transformation of data and implements it in an organized lexicon or grammar for programmatically transforming data. And what I mean when I say grammar for transforming data really relates to 2 characteristics of the dplyr approach. One, in a similar way to how verbs operate on nouns, dplyr functions operate on data frames. And 2, that dplyr functions use a consistent syntax, which minimizes cognitive load. So once you understand the dplyr approach with one function, it's much easier to learn new functions. So let's start with our first function. For anyone familiar with SQL or SQL, we'll be using a function that's very likely near and dear to your hearts that, like many functions in dplyr, has a counterpart starting with select. Select helps with narrowing down a data frame to just the columns that you need for an analysis. And this is done using the select function. And here you see the graphical framework for how select works. You provide the raw data as the input in the form of a data frame. And select will extract the columns of your choosing and return these columns as a new data frame. In the output, you will wind up with all the rows that you started with and the columns defined in the select function. So here you can see that select takes the starting data frame as its first argument. In this case, it's the COVID testing data frame. And after that, it takes any number of additional arguments that specify the columns that you want to pick. In its simplest form, those additional arguments will be the names of the columns that you want to select. Like in this case, we were indicating that we want to keep the MRN and last name columns. We don't want to bring any of the other columns out in the output. In very wide data sets, writing out the names of the columns you want just doesn't quite scale. So dplyr does have additional convenience functions that can help you succinctly identify the columns that you want. We won't go over these here together, but they can be found in the data transformation cheat sheet in the appendix of your course book and are available through tangential packages like tidyr and tidyselect especially. So let's look at this example. If you were to run the code you see here, the snippet of the COVID testing data frame on the left would be accepted as the input, and a subset of it, the MRN and the last underscore name column would be returned as the output. So first name and gender get dropped from the output. So let's do a little pop quiz. Which of these would successfully select the first name column from the COVID testing data frame? I'll give you 1 minute. Okay, I'm seeing a lot of answers coming in saying B. All right, I think that we have a group consensus. So B would be correct, and I'll quickly just explain why. So when we look through these options, A doesn't quite work because we didn't put COVID testing first. We're telling, in this case, select that first name is our data frame, and that's just not true. It's the column within COVID testing. B is correct because we put them in the right order. So COVID testing is the data frame, first name is the column we want to select on. The next one, C, is incorrect because, as we'll see, if you use a negative in front of one of your columns, you're actually telling select to take everything but this column, so everything except for first name. And for D, first underscore name with two capitals isn't correct because that's technically going to be a wholly different column. In this case, one that doesn't actually exist. So capitalization matters. Okay, so now that we have select under our belts for subsetting by columns, let's talk about how you might subset rows and observations using filter functions. So here you can see the graphical framework for how filter works. You provide the raw data as the input in the form of a data frame, and filter will use the logical criteria you provide in the function to identify the rows that are required. In the output, you will wind up with all the columns that you started with, unlike select, and just the rows defined in the filter function. So in filter, the first argument is again the data frame, for which you'll be extracting the rows that you want to filter on. Afterwards, we can input one or more logical tests. R then executes those logical tests on each row of the data set and returns all the rows in which the logical test is evaluated to be true. Rows for which the tests are true are returned in the output as a data frame, and the rest are removed. And the key point here is that the logical tests are evaluated row by row, one at a time, independently. So let's zoom in for a second on one of those logical tests. Generally speaking, they're going to take a form like this. There will be some column that contains the data you want to filter on, like in this, like the clinic name or the patient MRN. And then there will be a logical test. And what you see here is double equals, but you can also have greater than, less than, or much more complicated filter execution statements. And then you have the criteria. Equal to what? Greater than what number? In this case, we're saying we want to check to see if column name that we supply, such as MRN or clinic name, is equivalent to a certain criteria. So here's an example with the data set. So we supply the COVID testing data frame as the first argument. Afterwards, we provide one logical test. We're asking filter to return all of the rows in which the MRN is equivalent to 5000083. Filter will then go row by row, starting from the very first row, and ask, is the value in the MRN column equal to patient subject 83 for that row? And as you can see, it is not, so it gets an imaginary value of false. It then goes on to the second row and says, again, check to see if the MRN is a match. It's not, so it's also false, and so on for the third. But in the fourth one, we can see that we do have a match, so that gets a true. So the output of this function is a data frame with the same number of columns, but only the rows that resulted in a true based on the criteria in the function. And we see that here. Similarly, we can ask filter to return just rows that represent tests for a specific patient, such as last name equal to Stark. In this case, the first two rows result in true and so are kept. Note that when asking filter to return rows with specific text inside of it, we have to put that text in quotes, and it's got to be an exact match. Partial matches don't count unless you use something specific like regex to help you along, or certain other packages like stringer. In the previous slide, we were able to get away with not using quotes because the MRN column, I believe, is actually a integer double format, so we could actually supply that as a number. So what we've seen is that the output of a filter function is completely dependent on the logical operators in this green square. rows in which the logical statement results in a true are returned, and those that are false will not be returned. So the next question, of course, is how do you construct logical statements in R? And out of the box, R provides logical operators for pretty much every occasion. Here are just some of the important logical operators to know about, and we've already seen the double equals. There's also the quote like less than and greater than operators, and these each have their own equal to versions where you would just put an equals on the front or back to indicate if it's greater than or equal to or less than or equal to. And you can also use exclamation point with equals if you want to select rows in which a value is not equal to something else. And so this bottom one here, it says not equal to reads, look for rows where x is not equal to y. So let's do another pop quiz. Which of these would successfully filter the COVID testing data frame to test with a positive result? So start that timer. Some of those results coming in. Looks like everyone is pretty consistent. Okay, I think that's good enough. So let's see. So that would be correct if you said C. So let's go through again why C is correct. So A is not correct. I'll turn off the timer. All right, A is incorrect because we're setting result equal to positive without quotes. And again, the filter that we have is on a column where everything is a character string. So we need to actually say that this is within quotes to signify the string value. B is incorrect because this only has a single equals. So again, a single equals, you can think of that more as saying that you are assigning a value to something, and that just doesn't quite work within filter. Whereas in C, which is correct, you have double equals, and this is saying that we want to check for equivalence. So result equals equals positive is correct. And D isn't correct because we have the order flipped in which we're saying positive equal to result, where result is actually our column and positive is the value we want to filter result on. All right, so now we're going to up our skill level a bit and talk about data pipelines in a much more literal sense. So this is the pipe operator, this percent greater than percent series of values. The pipe operator is a powerful tool used throughout dplyr and will be one of your best friends in data analytics. So the pipe operator is this percent greater than percent symbol. It's a tool that we use to sort of glue the pieces of our analysis together into one consistent pipeline. The pipe operator does something very simple, but at the same time, incredibly useful. It takes the product of whatever is on the left and puts it into the function on the right. You can think of this sort of as that's taking those values and carrying it through your code pipeline. So for example, the two lines of code in the gray box here do exactly the same thing. And there are a whole bunch of reasons as to why this is a useful thing, including that it turns computer code into something that is almost legible as human language. And you could essentially read the second line of code as, say, take the COVID testing data frame and then filter on pan day less than or equal to 10. And the advantage of this is even more clear when performing a more complicated analysis such as this one. You can imagine that if you're doing multiple operations, you're going to be doing even more of this. Over here, you can take this code, which is essentially illegible, and transform it into the code on the following slide. And I see Sylvie. Yes, that is a great point. We have a brief shout out to that, I believe, shortly. So if we instead use the pipe operator, the code here is far more succinct than the previous slide. It's essentially self-describing since it's written in a sequential, essentially human-readable format from the top down. So the pipe operator allows us to form chains that accomplish complicated data transformation operations, but which are made up of very simple, self-contained building blocks. So we're able to make something complex by joining together many simple elements that are easy to understand in isolation. An additional benefit is that it makes it easier to actually write code because it's much easier to translate our thoughts into an analytical pipeline. And as people are saying right now in the chat, the new pipe is this. I actually don't remember the name of that character, but it's the tall bar with the greater than symbol. Comes with base R out of the box. I believe you need to have R version 4 or 4.1. Thank you, the pipe, native pipe. 4.1 or greater installed, so it won't quite work with 3 and below. So you might want to just be on the lookout for that. But you will see this a lot more in the wild, especially as time goes on. There are some corner case scenarios where things don't work with it that do work with the Magritter, which we'll call the Magritter pipe, which is the percent greater than percent symbol. But generally, they're almost always equivalent, which I believe Philip mentioned. 91%. Can you elaborate on that, Sylvie? Anyway, you can elaborate, I'll keep going. Okay, so next pop quiz. Which of the following is equivalent to the set of code you see here? Data pipe select column name. Okay. And yes, that is what Philip said is correct. The period, which is often used, won't work with the Magritter pipe, but there are some workarounds, I believe. All right, so I see that we have a consensus once again. Great job, everybody. If you said D, that is correct. Let's again just briefly talk through why that is. So let's see. So A is incorrect because in this instance, we're calling data itself as a function, putting it before the parentheses, and that's going to be actually really confusing because out of the package, data is actually a function. I believe it pulls different data sets for you or as a helper for that. So that's going to not work well with this. B is incorrect because, again, we're actually calling data as a function just inside of select. C is incorrect because, again, remember those ordering constraints. We actually need to call data first before column name. Here we're saying that we want to select the data column out of the column name data frame, and that isn't correct because column name is not a data frame. And lastly, D, which is correct, which is that we're saying we want to select from the data frame and take out column name. Okay, so now we've discussed a couple of ways of manipulating existing columns, but what about if we want to add or create some new ones? And this is where the mutate function comes in. So with mutate, you're going to feed your original data frame, and it's going to return the same data frame with one or more new columns tacked onto the end based on the calculations that you specify. And the syntax is going to look familiar to the functions we saw earlier with a small change. As before, the first argument is the original data frame, so COVID testing, and we're showing it here with that pipe operator. So remember that it's actually being piped into the first argument of mutate. And then you specify the name of the new column, that you want to add, and you type an equal sign, and then you write a calculation. This calculation will be run on each row of your data frame, and the result will populate each row of the new column. Note here that we are supplying the single equal sign. I mentioned this earlier, that we are signifying assignment here, the assignment of the values in this calculation, instead of the double equal sign, which is, again, an operational filter check. Or equivalence check, sorry. So in this example, right now the TAT columns are coded in hours. If we wanted to calculate the TAT column in minutes, we could use something like this. We take the COVID data, we take the COVID testing data set and pass it into the mutate function. And then the mutate function will do three things. One, will make a name for our new column, C underscore R turnaround time. Yes. C underscore R underscore TAT underscore mins. That's going to be the name of our new column. We put that equal sign to say that all the values in this new column are going to be equal to this calculation. Then we define our calculation. The calculation whose result will be populated there. And in this case, we can directly reference the col underscore REC underscore TAT column and multiply by 60. All right, so let's do your term number 1. So open up the transform.qmd and work through the first set of exercises and click yes when you are finished.
So I don't think we have a place to click yes. Instead,
Sorry about that. Yes, just say done. Just type done in the chat when you are finished. My apologies. So we're about halfway through. See a few dones. Okay, so with this last minute and a half, I'm just going to say that we don't quite have time for me to live demo. But if you're running into any issues, please put questions in the chat. And you can also look under the solutions folder if you're struggling and need to reference how it is intended to work. Okay. All right. So again, just post in the chat if you're having any issues, but you can always reference the solutions if you need help as well. So let's continue on. Okay, let's get into another slightly more complicated thing that dplyr gives us. So let's say we want to perform certain calculations or manipulations on cohorts or groups of data. How would we do it? To answer this question, there's a way to tell R that data itself is grouped, and that is by using the group by function. It's a challenging function to master, but extremely powerful. It's one of those tools that once you understand how to use it, you cannot possibly go back to living without it. Group by allows us to take a data frame and break the observations up into several groups. And then we can look at those groups separately. So through color coding here, you can see how we've separated out the red, green, and blue. So here's your basic code to construct for grouping. We pass a data set using pipes to the group by function, and then we specify the variable that we want to define the groups. Now, using the data set we have, let's take an example, looking at grouping by pan_day. We can see that the resulting object has just over 15,000 rows that are broken up into about 102 groups. This means that there are 102 distinct pan_days. Some of these groups will have a single observation. Many of these groups will have more than one observation. We can also group by more than one variable. Sorry about that. We can see that the resulting object has still that 15,524 rows that are broken up into 2,526 groups. This means that there are 2,500 distinct combinations of pan_day and clinic_name. Again, some of these will have a single observation, many of them will have multiple. Let's see. Now let's talk about how we can leverage group by in addition to some summarized calculations. Summarize the function allows us to take a variable or column of data and apply a function or multiple functions to all of the observations in a table. So using summarize, we can return a single value or summary. To make our take summaries of our data, we use the following code construct. We pass a data set using pipes to the summarize function. And within the summarize function, we provide a variable name and set it to a calculation or value that we want to define. Let's take a look at a simple example using the summarize function to count the number of orders in a data set. Note that to simplify the data frame, we first will select two variables and include only the first 4 observations. That's what head is showing there. So we're saying take the head versus tail, so the top of the data frame versus bottom of the data frame, first 4 observations at the top of the data frame, and use the n function, as you can see here on the right side of order count, to return the number of observations or rows for a data frame. So within this summarize function, what we're doing is almost in a way similar to mutate. We're saying we're making a new column, a new value called order underscore count equal to a calculation, which is n, which is just the number of observations of the rows in the data frame. So what comes out of this, all in all, is 4, because we only have 4 rows. Because again, remember, we took the top 4 rows of the COVID testing data set, not the whole 15,000. And you can take any number of additional summaries within the summarize function. These will all amount to adding new columns to your output. So here we're still taking those top 4 rows, and we're going to add another summarize column called day underscore count using a function called n underscore distinct of pan day. And unlike order count, which looked at how many rows or observations are available within the data frame, day count is going to look for the number of distinct values of pan underscore day. So whereas order count came out with 4, day count comes out with 3 because there are actually only 3 distinct values here in pan day: 4, 7, and 8. 7 is repeated, so we don't have 4. In the output, we have 3 instead. Let's see. So we can expand on this one step further by looking at the whole data set, not using just the first 4 rows, and calculating orders per day. I like to note that the order of column creation kind of matters here. So in things like mutate and summarize, because while we're executing this code all at the same time within our console, each step is processed in sequence. So orders per day takes the created order underscore count column and divides it by the other created day underscore count column. So we see in the total that the original, we have the original 15,524 orders, the original number of rows, 102 unique days, and now there are 152 orders per day. So going back to using group by, so just like we can supply summarize to a full data frame, we can supply summarize to a grouped data frame and evaluate each group separately. So the combination of group by and summarize can be extremely powerful. So using summarize and group by, we can count how many total rows there are. So the original without group by was the 15,524. And then by adding group by, we count how many rows there are for each type of pan underscore day. So here saying COVID testing, group by pandemic day, summarize the order count, so the number of observations within each pan day, and that will give us the number of observations within. This is also, again, I don't think it shows here, but this is the first 4 rows. So okay, so I'll give you guys 3 minutes to do your turn number 2. So work through the exercises, and sorry about this. Don't click yes. Say done in the chat when you are finished. See a good question here about ID and .by parameters. Yeah, so this is actually really great, and hopefully not going to confuse too many people here, but it is worth mentioning that in recent versions of the tidyverse and dplyr, I'd say within the last year, maybe 2 years, they did add a few new parameters. .by essentially functions the same as group by. So if you're comfortable looking into the documentation, you can actually use .by and get around having to use the combination of group by and ungroup. That I typically tend to do, but it does require more up-to-date versions of the packages themselves. Okay, and as a reminder, if you're having any difficulties, post in the chat or reference the solutions QMD. And we will round out this session. Okay, so let's do a quick recap of what we covered up to this point. So we can extract data we are interested in from datasets using select functions. We can then hone in on specific observations of interest by filtering with logical criteria. And finally, we can create and shape data using mutate functions. By using the pipe operator, we can combine transformation steps to make our code easier to read and even easier to type. And one thing to note that if you're comfortable using hotkeys within your RStudio environment, Command Shift or Control Shift M is usually an easy way to put the pipe operator into your code, and usually saves a bit of time, a little bit of some keystrokes. And we can use the group by function, or and if you're engaged with the chat, you can see that you can use the .by parameter within a number of these different tidyverse functions to group your data together and to calculate summary statistics within those groups. So what else is available beyond this? dplyr and the expanded tidyverse have just an absolute ton of functions for you to explore, but just to give you a few. There's a range, which lets you arrange data by specific columns, such as numeric or date values. You can add rows to your data using add_row. You can bind columns together, and although not shown here, you can also bind rows together if your data is shaped correctly. For those familiar with SQL or SQL, all of the different join functions are available here and are extremely powerful. Left join, right join, inner, outer, all available. And as Stephan mentioned before, there is a dplyr cheat sheet as well available to you, which has all of these and more. And I encourage you all to look at the dplyr website, which goes through a number of these as well. So dplyr is, again, just one of a number of tidyverse packages that work harmoniously. You'll call a lot of these in conjunction with each other to provide you with all the tools to shape, analyze data for consistent, reproducible, and succinct data pipelines. And here are just some, including tidyr for tidy data operations. We've mentioned before lubridate for date handling, stringr for character and string processing, the per function or package, which has a widespread of mapping functions, and dbplyr for database plyr, which is a database for database operation similar to dplyr, but tailored for database manipulation. Okay, and I will send it back to Stephan.
Thanks, Rich. This was a super quick introduction into dplyr. I think you did a fantastic job. Now, unfortunately, we have less than 10 minutes. We actually have 6 minutes left to go through the rest of the course. So I don't think we're going to be able to to really talk about dashboards, I will very briefly mention that an important way that clinical information is presented is not in static reports that need to be built manually, but interactive dashboards. So dashboards are all the rage in healthcare in general. So this is because dashboards visualize key data on demand, and this allows your SMEs or subject matter experts who are not trained in performing analytics to quickly find essential information to do their work. Fortunately, there are some pretty cool tools and are available that you can use to build dashboards, and one of them is a package called Flex Dashboard, which actually acts on R Markdown, not Quarto, and can turn an R Markdown document into an interactive dashboard. So we'll have the GitHub repository with example exercises on how to make dashboards described in this your turn exercise, but we unfortunately don't have time to go into it. One other thing I wanted to mention is that I wanted to highlight our two packages to really make it super easy to add some interactivity to your dashboards. So one of them is called Plotly, and Plotly can take a ggplot object and turn it into an interactive plot that allows you to hover over it and zoom into it and do some other neat things. And it is super easy to turn a ggplot into a Plotly plot. And there's an exercise in the repository that'll walk you through how to do that. The other thing I want to turn out, the other package I want to turn out is called DT for data table, and DT makes data frames interactive. So if you have a data frame that you're posting on a dashboard and you want to make the tables interactive, similarly to the data viewer that we looked at in the RStudio IDE, you can use the DT package and the data table function. So data table just takes the one argument that's a data frame, and then outputs an interactive data table that can directly be displayed on your Flex dashboard. And you can add styling easily. You can add a theme by adding just one line into your header of, remember, Flex dashboards use R Markdown documents. So R Markdown documents, just like Word documents, have headers. And by just adding themes serially, you can theme your dashboard in a certain way. So here, just a very quick recap. R Markdown documents can be turned into interactive dashboards with Flex Dashboard. You can turn ggplot objects into interactive plots with Plotly, and you can turn data frames into interactive tables with DT. What else is there? There's more interactive charts that you can look at by going to gallery.htmlwidgets.org. If you want to build a full-fledged interactive web application in R, take a look at Shiny. Shiny applications can be deployed on shinyapps.io for free. Or you can deploy your own Shiny server, which is possible to do for free, but it involves some elbow grease to get this to work. Or you can use a paid POSIT product called RStudio Connect to deploy dashboards and web applications. So we're at the end of the workshop, and we really encourage you to install R and RStudio on your computer. If you want to practice more, and if you want to go over some of the materials that we had to unfortunately skip, please go to our GitHub repository and you can click the green button labeled code to download the whole repository as a zip file, so you have all the files that you had in the Posit Cloud environment on your computer. We're also this isn't live yet. This is going to be live by the end of the day. We're also going to have a website that comes with the course that's going to walk you through how to set up R on your computer and RStudio, and it'll point you to how to walk through the exercises from today if you want to do it by your own, which we highly recommend. We also for further reading, we highly recommend the R for Data Science book by Garrett Gromund, Hadley Wickham, and Minich and Kaya Rundell. This is the standard introduction to data science with R, and the entire text is available for free at this address. This is the second updated edition. Each chapter has a lot of exercises, which I encourage you to do. So what should you do when you get stuck? So we used to recommend the strategy of Googling and looking at the cheat sheets. We no longer recommend this. Instead, we strongly recommend that you use generative AI like ChatGPT as a tutor when you're working through exercises. So you can go to ChatGPT. You don't even have to log in or make an account. Even the free version that you can access without making an account is now good enough to help you troubleshoot your R code if you're working through, the R for Data Science book. One word of caution: when you use a public LLM, never enter any protected health information, any personally identifiable information, or any proprietary or confidential institutional information. My rule of thumb is, if you have to think about whether it's okay to type something into an LLM, don't do it. But for learning and how troubleshooting your ggplots and the exercises, it is absolutely a phenomenal tool to help honing your skills. At this point, I want to thank our TAs. Thanks, Alex, Ezra, and Roni, for helping out. And please, and thank you for joining us and for sticking through 3 hours on a Monday morning on the East Coast, and there may be all kinds of different times for our visitors who are joining us from all over the world. So thank you very much. I would really appreciate if you could take a few minutes to complete the post-course survey. Even if you don't have to fill out the entire thing, but we're taking feedback extremely seriously, and if you've noticed that something was missing or was incorrect or was something should have been covered, we do look at this very closely, and we continuously improve this course. So again, thanks. Thanks for sitting through this, and we will have the course website up by the end of the day. Maybe there's going to be a way to share the link to the course website as part of a mailing for the R Medicine conference. We'll figure something out, but you'll have the link to go back and do those exercises that we